Hello, and my name is Stephen Petru from the Florey Institute of Neuroscience and Mental Health in Melbourne, Australia. I'm going to talk today about ASOs and gene therapy in the genetic epilepsies. I'd like to thank KCN2Q2 Cure for the invitation to speak. So I want to talk about gene therapy broadly in pediatric neurological conditions, but also and particularly give an example around how gene therapy can be used in developmental and epileptic encephalopathies from work from my own laboratory. But first, just want to briefly touch on the emerging genetic architecture in developmental and epileptic encephalopathies. And fundamentally, there's lots and lots of ways that we can break a gene, and then there are different ways that we need to think about for delivering a therapy as a result of those ways that the gene is broken. Probably the thing we're most familiar with is de novo single nucleotide mutations that arise some point along the journey from formation of the gamete up to and beyond, up to the different stages of the zygote and embryo. We can see gain of function, a loss of function emerge. We can even see mutations that give both rise to a loss and a gain of function. We can see copies in the numbers of a normal gene, both deletions and duplications. Copy number variation is one of those areas that many of you may have heard about. Inheriting through autosomal recessive mechanisms, the so-called compound heterozygotes, where you've got a one from each parent, can give rise to disorders, and probably more common than we've thought as a architecture of some DEEs. X-linked mechanisms, and there's whole unknown areas. What does the non-coding DNA do? How might it contribute to disease genesis is largely untapped. We know about mosaicism. Not every cell has the same genotype, and the way that unfolds can alter the expression and time course of different disorders. And epigenetics is another area where intergenerational changes in the genome can impact disease as well. Each of these different mechanisms requires a different thinking around therapeutic delivery. We know within the single gene DEEs that it's been a very rich area due to pioneering work by many groups. I'm not going to belabor this slide, but it's important to know that for different syndromes, there are lists and lists of different genes emerging, but there is very clear clinical and genetic heterogeneity as well. So that's something to bear in mind when you are looking at the natural history of many of these disorders. What is gene therapy? I think there are different definitions used by different groups. I use a very broad definition as a treatment that directly operates at the genetic rather than the protein level. And that's not only limited to delivery of a gene through a virus. It can be an siRNA, or it can be an antisense oligo. Gene augmentation therapy is to deliver a new gene to the body, most commonly with a virus. We can use different technologies to switch genes on. We can turn genes off or down using siRNAs or antisense oligos. And then probably the most promising area in terms of the impact it might have is to do in vivo gene repair. And technologies such as CRISPR hold that promise. There's issues still to be overcome, but that was one way of affecting a permanent fix. Where are we now? I'll show you in a second some of the history of where we've come from, but there are about 20 new gene therapy products that are currently on track for FDA approval by 2025. So lots of active programs, most commonly taking advantage of either single dose viral delivery or antisense oligos. And we'll be seeing that's a real acceleration from where we're currently at. And this is in the neurological space, pediatric neurological indications mostly, not more broadly in other disorders. And there are multiple trials underway, including many of the ones that we're interested in. This is a slide taken from a recent review, and the acknowledgement dropped off, unfortunately. It's from the group at UCL by Dr. Ng. And it really shows nicely from 1998 to about 2020 regulatory approvals of different products, including ASOs, different viral modalities, and siRNAs for a different series of neurological disorders and neuromuscular disorders. So you saw an example of the various programs that are currently underway seeking to deliver gene therapy in a host of neurological disorders, in particular in pediatric cases. But how do you go about thinking about what therapeutic modality you might choose for a specific disorder? If you look in the general toolbox of gene therapy, antisense oligonucleotides are one particularly important class of therapeutic molecule for epilepsy and developmental and epileptic encephalopathies in particular. These are artificial DNA molecules that have been chemically modified. They're chemically modified so that they last longer in the body. They're chemically modified so that they bind stronger to the target that they're meant to bind to, and I'll talk about that in a minute. That's important because if you can bind them stronger, you need less of them. And then the third point, which is, are they going to cause any toxicity? Are they going to be well tolerated? The chances are better if there's less of them in the body in general. This is a very important class. You may have heard of the application of these clinically in spinal muscular atrophy with the Spinraza molecule. Ultragenyx is moving programs forward in Angelman syndrome and other disorders. So a very, very important class of molecule. They are interesting because they don't just do one thing. They're not a one-trick pony. ASOs have various modes. They're overviewed in this slide. And I won't go into detail in each of the different modes. Suffice it to say, in the top sample there, they can alter the way proteins are made. And in the second slide, you see it a different way. They can alter how proteins are made. They can change the way RNA is used to make proteins, and you can RNA is like a blueprint, and you don't have to use every part of the plan. You can miss certain parts of the plan, and ASOs can be used in that mode. ASOs can be used to interact with natural antisense oligos. There are natural antisense oligos in the body already that do things to alter gene expression. An antisense oligo against the natural version of that can block that effect, and we know about examples in Dravet syndrome. That company called OPCO is trying to use that approach, and I think there's also a program for SYNGAP1 that's trying to use this approach in order to block the natural blocker, thereby increasing the amount of desired protein. Above on the right, I'm showing other examples of how antisense oligos can enhance the amount of protein, particularly important for loss of function disorders. And you also another way on the bottom, in the middle there, showing how they can be used to modulate RNA to remove aspects of the RNA that are causing it not to produce enough protein. And by removing those elements, you get more protein made, and this is the basis on which the company Stoke Therapeutics is employing to produce a therapy for Dravet syndrome. And there's also ASOs are very primed and poised to interact with microRNAs. MicroRNAs are naturally occurring RNA molecules that can interact without mRNA and cause different levels of expression of target proteins, and antisense oligos are another way that can be used to interact with those and affect a desired mechanism of action. One of the modes I want to talk about today that's relevant for the example I'm going to give, and it's also relevant for how you might apply this technology to KCNQ2, and it's really one of the most well-trodden modes of ASO action, so-called GAPMA. I'll tell you why in a sec why it's called a GAPMA. But in this mode, the ASO causes degradation of the target RNA, and this RNA could be a bad player, it could be a mutant piece of RNA, or it could be wild type, or it could be both. The antisense oligo binds somewhere along here, shown in the top left, binding to a part of the gene. Once that binding occurs, it then triggers the activity of an enzyme called RNase H. And this enzyme detects the interaction of the ASO with the target mRNA and causes destruction of that RNA. Now, that ASO needs to be in a particular structural configuration called a gapmer, where there is different levels of chemical modification on the wings of the ASO versus the core. And I've just shown that here on the bottom, where you've got the blue on the wings and the green in the core. And when that type of ASO binds the RNA, it gets destroyed. And that's very useful in dealing with gain of function or dominant negative when you take one further step that I'll talk about in a second. So you can see here, if you have in the case of KCNQ2, where you often have dominant negative effects, where the mutant allele not only doesn't function normally, it also, when it makes protein, the mutant protein interacts with the wild type protein and further disrupts the ability of the channel to work. If you could destroy the mutant protein only and leave the wild type alone, you would relieve that dominant negative effect. And you could do that by making an antisense oligo that binds the mutation specifically, and it targets that mutant allele at the bottom for destruction. You can see here the red part's the mutation. That red box isn't in the wild type. The ASO can detect the difference between the wild type and the mutant, and it binds. And when it binds the mutant, it causes a stronger reduction. It'll still bind the wild type a little bit, but this mismatch of this red region will mean the RNase H response isn't as strong in the wild type as it is in the mutant. And you can often, with a judicious choice of a region and a bit of luck, you can achieve up to 100 times selectivity. So this method will let you destroy 100 copies of this messenger RNA, and with only one copy of this one destroyed, which would be a great situation for something like KCNQ2. We can do this in a different mode because if it's a mutation-specific antisense oligo, it will only be useful for those patients that have precisely that mutation. And that limits the application of it and makes it more difficult to get the resources together to develop a specific ASO for a specific patient. You can target a common change in the RNA and DNA that's in phase, we call, with the mutation. So it's on the same copy of the gene. Remember, we have two copies of the gene, one from each parent, and the mutation resides on one of those in disorders like KCNQ2. If we can find a so-called single nucleotide polymorphism, or a SNP, that's in phase with the mutation, the ASO can interact with it and cause degradation, more specific degradation of this allele versus that. Thereby, and these SNPs are common in the population. So you'll find many, many more patients that have got this SNP, and if they're lucky, the SNP is in phase with the mutation, and then this ASO will target them. And it's not as general as targeting KCNQ2 overall, which we don't want to do because we know that the too little like KCNQ2 is not good, and destroying both copies isn't going to be therapeutic. So we do need to go that extra step for KCNQ2, and this is one way that we can achieve that. So just if you take a step back now and think more generally about how you might, what ASO might you use for different genetic mechanism. Well, a gain of function disorder, you might just want to reduce the gene. And that's happening for SCN2A, for instance. And I'll give an example of another potassium channel today where that's the same situation. It's the potassium channel just works too hard. You want to get rid of it. You just reduce it with knockdown with a gapmer ASO. If it's loss of function mutation, the mutant causes a protein to be made that doesn't do enough. What you might want to do is make more of that protein. so-called upregulating. And then, as I mentioned, in the ASO modes, there are various ways you can do that, either by manipulating the amount of messenger RNA or manipulating the efficiency of the production of protein to try and counter the effects of the loss of function. As is the case with KCNQ2, where you have dominant negative, or in those cases where the mutation has a strange effect. Some of the effects on the protein are gain of function, others are loss of function, and it's clear that that mutant allele shouldn't be around. Not only because it's causing dominant negative interactions, but because it's causing other pathological effects. You move this allele-specific knockdown, such as just described. And then for recessive genetic disorders, there's a host of other ways targeting microRNAs, targeting splicing, or other modes. So you can see ASOs are fairly well poised and have quite a few options available to you depending on the precise genetic mechanism. The example I want to give today is epilepsy of infancy with migrating focal seizures, or EIMFS. Like KCNQ2, this is a potassium channel. The patients are very severely affected, onsets early infancy, frequent seizures called migrating because on the EEG they move from one EEG region to another. These kids do fairly poorly, profound global developmental delay, no effective treatments, very high mortality, lots of comorbidities with these kids, unfortunately, very commonly caused by KCNT1 mutations. This channel carries a potassium channel, potassium current. It is activated by sodium and voltage, and it works in concert with the sodium channels to form and shape action potentials. And what we do see when we study it in an expression system is that mutant channels produce more electrical current than wild type channels. And this is seen unequivocally across the board for all the mutations that we and others have studied. So it's very, very clear to be a gain of function. So in thinking about a therapy you might use to tackle this, knocking down the mRNA, knocking down gene expression is what comes immediately to mind. The first thing we did here was to produce an animal model, and it was clear in humans the disorder is heterozygous as a de novo condition, autosomal dominant. In mice, we don't see a strong phenotype in the heterozygotes, but we do see it in the homozygotes. Here you can see in red in the survival curve that all the animals are dead by about 100 days of age, with death starting to occur around day 35. Wild type and het animals have the identical survival, and we see no deaths after about 6 months. Animals are smaller. The homozygous you can see with the arrowed animal and shown on body weight on the right, runted. The homozygous animals show spikes, and you can see up to 1,500 spikes per 24-hour period on EEG, and they also show a number of spontaneous seizures in comparison to wild type and heterozygous. So we do see electroclinical phenotype. We do see this a cognitive change as sort of modeled by this ability for the animals to build nests in their home cage. When you introduce tissue paper into the home cage, most animals within about 24 hours will develop this little round nest at the top here, and they sleep and rest and spend a lot of time in these nests when they're not foraging for food. The homozygous animals can't construct a nest, and the tissue paper just stays flat. And you can see here, this is a homozygous animal that's had these tissues for a couple of days, unable to build a nest. We can quantify the quality of the nests here. Wild type heterozygotes score almost perfect levels of 5. Homozygous animals are around 1. Big difference in this ability, and this is a sort of a aggregate cognitive and sensory ability for these animals to be able to do this, and providing a benchmark on which to say, if we do have a therapy, can we see correction of the seizures, but can we also see improvements in these cognitive domains? And that's something that patients and physicians alike are looking for in their therapies. We did look at other phenotypes, motions, movement in locomotor cells, time spent in light dark box areas, elevated plasmase. I won't go into the gory details of those. We're not saying that there's an anxiety or not phenotype in these animals, but we are saying that there are differences in them. They move more, they spend more time in the dark box, and they spend more time in the open arm of the elevated plus maze. And this is just summarized here to show that there's a clear battery of phenotypic changes in these animals that we can use as a baseline to test and assess the performance of our therapy. With respect to the therapeutic mode that we selected, we chose ASOs. We chose them because there are different modes that ASOs can work in terms of they can cause upregulation, they can direct splicing, and they can also reduce RNA through this so-called gapmer mode. And it's called a gapmer because the chemical modifications of these non-natural nucleotide bases are done in a way that the wings of the molecule, this is a 20-mer oligo, the wings have got a different chemical modification than the core. This type of ASO, when it binds messenger RNA, directs the RNase degradation of that bound piece of RNA. So this complex of this gapmer ASO bound will cause destruction of that RNA. As shown here, we use these ASOs because the chemical modification of the nucleotides, and when you make oligos out of those, have higher affinity and stability, so they bind tighter, they last longer in the body, and they are tolerated better than other chemical modifications. And this is all the hallmarks of a good drug. You don't have to put massive amount in, so high affinity means you can use less drug. Stability means that for an invasive administration, which is this intrathecal, you can do this less frequently, 2 to 3 to 4 times a year, depending on the half-life, which can be measured in the months, and you can make it well tolerated, so you don't get hybridization-independent adverse effects. We collaborated with Ionis Therapeutics on this, and there's a paper on bioRxiv. If you're interested, if you search for my surname Petru and KCNT1, that'll come up. ASOs were screened, and we can look at potency. So here, dose response curve, and we can understand how much ASO we need to deliver to different aged mice to see a 50% or an 80% knockdown. It's specific. KCNT1 gets knocked down, the related gene KCN2 does not. So we can make it very, very specific because it's a hybridization mechanism. We can select a unique sequence in order to achieve specificity. We then ran through a battery of tests in the same mouse model that I just described to you. ICV injection was given, and some pre-video monitoring was done to get some baseline. Between days 40 and 45, ICV ASO was delivered. Surgically, you can see here with the skull shown where we do the ICV injection. And then we did a whole bunch of the same battery of tests that we said: locomotor cell nesting, electrocorticogram, light dark test, and elevated plus maze. ASO gets everywhere. There's an antibody here that picks up the backbone of the ASO. Non-treated shown dark, ASO treated shown very good distribution, including subcellular. We know, and one of the good hallmarks of a good antisense oligo program is that the knockdown of the target has low liability for toxicity. And here we've knocked down, essentially eliminated the KCNT1 gene through a massive dose of the ASO. And we see no difference in the wild type behavior following reduction. This is consistent with findings in the knockout animal that have got mild phenotypes, and consistent with some of the human studies where there are many people in Nomad database that are missing an entire copy and are, for all intents and purposes, normal and unaffected. Here we dose the animals with 3 different levels of the ASO, including a control. You can see in red the uninjected animals dying and all dead by about 100 days. In orange is the control ASO. You often see some pharmacological effect of giving the ASO, but you see clear gene dosing here as we give higher levels of ASO, 50% knockdown, this light purple, darker purple, 80% knockdown, and the top was a massive dose of 500 micrograms that was greater than 95% knockdown. You can see a single dose, all of these, and you can see as the ASO gets metabolized, the phenotype re-emerges. And we also started to see seizures re-emerging. So clearly we can stop the event. The ASO is protective. As the ASO wears off, the gene comes back. You start to get the mutant allele being re-expressed, and the disease phenotype re-emerges. Seizures, spikes, abolished. This is untreated animals, and these are the treated animals. No spiking on the EEG. And here, looking at pre and post ASO, we can see complete abolishment of seizure activity at 50% knockdown, at 80% knockdown, and greater than 95% knockdown. No seizures in those treated animals. Control ASO animals, no protection from seizures. And really remarkably, we saw a reversal of this cognitive effect of the nest building. We can see the top animals ASO treated, mutant animals building nests, control ASO, no effect, and this is summarized here in this graph. Untreated animals, a control ASO, poor nest building, all the three different doses giving a very similar improvement in this ability to build nests identical to wild type, showing that we can get both cognitive and seizure protection. So before I acknowledge the people in my group and my collaborators, I'd just like to hope I gave you an overview of some of the ways forward that could be very readily applied to KCNQ2, then to show you that there are many programs already moving forward for other genes using very similar antisense oligo approaches. And I think that an allele-specific program is underway for KCNQ2. And I think there's great reason to be optimistic. It's always important in this area to be cautiously optimistic. But I think there's everything's happening, and there's numerous examples. The more success and more activity there is in other programs using antisense oligos, the greater chance that programs that are starting in KCNQ2 will always likewise benefit and be more successful as a result of that. Small molecule programs, we know of various of those happening. There's been some activity recently in that space that I think we need to pay attention to. So I think, despite the challenges that I realize everyone is facing who have children or loved ones affected by this disorder, there is a steady and continual efforts happening globally to try and deal with this. I just want to thank people in my own group who did the work, especially towards the latter part of the talk around the KCNT1 project on the left. Liseth Burbano, Melody Lee, Umish Nair, Nikola Yankowski, Snezana Maljevic, Chris Reed, and Kay Richards, as well as my collaborator Paul Thomas. From Ionis Pharmaceuticals, Frank Rigo, Payman Yafar Nahad, and Frank Bennett, and funding from NHMRC, Mitchell Foundation, and Praxis Precision Medicines. Thank you, everybody.